Let's talk about how DNA gets replicated. And in order to understand that, it's important to review the structure of DNA. As you probably already know, DNA is made up of two strands, and the unit that make up these strands is a nucleotide. A nucleotide is made up of three components. The first is a sugar, or more specifically, deoxyribose, which I drew here on top. And in our central diagram, it's the same except for I left out the OHs and Hs so that it's more clear. And we label the carbons. This carbon is 1', this one is 2', 3', 4', and 5'. The second part in a nucleotide is a phosphate group. That's these purple P's that connect the 3' carbon and the 5' carbon of two nucleotides. Of course, the lines represent bonds. And the third thing we have in a nucleotide is a nitrogen base. And that's those, the A, T, and the C, and the G. So this, for example, would be one nucleotide. This would be another nucleotide. And when DNA is ready to be replicated, an enzyme will come along. This enzyme is DNA helicase. And DNA helicase is going to unzip or cut our double-stranded DNA into two separate strands. And here's what we're going to get. This is our original double-stranded DNA. At the end of replication, we're going to get two pairs of DNA. And each one of these pairs is going to have one strand from the old DNA and one strand that's newly synthesized. And this concept, this idea, is called semi-conservative replication. And semi-conservative replication just tells us that the new pairs of DNA that we have will have one strand that's from the original strand. We call that the parental strand. And it will have one strand that's newly synthesized, and we call that newly synthesized strand the daughter strand. And which enzyme is going to put together DNA? That enzyme is called Dna Polymerase. Dna Polymerase is the main guy here who's going to synthesize and put together our new strands of DNA. And it's important to understand how DNA polymerase works. So when DNA polymerase elongates a particular strand of nucleotides, it can only add nucleotides to the 3' carbon of any other nucleotide. So for example, it can add a nucleotide right here. It cannot add a nucleotide to a 5' carbon. So if this strand here on the left was being elongated by DNA polymerase, it would be elongated in the 5', 3' direction, and that's this way. Let's look at the strand on the right. Again, DNA polymerase can only add a nucleotide to the 3' carbon, but it can absolutely not add a nucleotide to the 5' carbon. So if DNA polymerase were elongating the strand on the right, it would be doing so in this direction. So the very, very important take-home message from here is that DNA is synthesized in the 5', 3' direction.